Good morning, students. Welcome to the biology class. We are going to continue with our same chapter, Cell, a Unit of Life. In our previous biology class, we discussed about the prokaryotic cells feature, in which we have already discussed about the outer covering of a prokaryotic cell. And today we will discuss mesosomes. Mesosomes, it is a structure which is formed by extension infolding of plasma membrane into the cell in the form of vesicles, tubules and lamellae. So this is the extension of the plasma membrane, and that extension may be in the form of vesicles, that is in the spherical form, tubules or the lamellae. What are the main functions of the mesosomes? Mesosome increase the surface area of transport system of bacterial cell. Bacterial cells means your prokaryotic cell. They play a significant role in replication of DNA. Replication of DNA means the forming the copies of DNA. From one DNA, two dotted DNAs are being formed and they go on further. So replication of DNA means the formation of the copies of the DNA. They helps in the formation of cell wall during the binary fission, because binary fission is a mode of reproduction in bacterial cells, means in your prokaryotic cell. And the very important function of mesosomes, they helps in respiration, secretion and enzymatic content. The mesosomes are the structure which is formed by extension and infolding of plasma membrane into the cell. You can see in this image this infolding, where I am giving a pointing out the structure. This is the infolding of the plasma membrane, and that infolding of the plasma membrane, that extension of the plasma membrane is known as mesosome. And when this extension is going to be divide, or it is going to be increased, it will increase the surface area, it will divide the cell, it will replicate the DNA, it will helps in the respiration, it helps in secretion. These are the important functions of mesosomes. So very important question regarding the mesosomes in your NCERT exercise is, what are mesosomes? Mention their important function. The next is your chromatophores. Chromo means colored bodies. So these are the internal membrane system containing pigments. Pigments means wherever there is some color structures are there which give them a color bodies. So they are extensive in the photosynthetic bacteria. The example of photosynthetic bacteria is sulfur bacteria and cyanobacteria. Cyano means blue-green algae, and the color of blue-green is because of the chromatophores. So these are also some extensions like that of the mesosomes. They are the internal membranes, but they contain the pigment and they give a color to that of the bacterial cell. Next, and the very important of the prokaryotic cell is genetic material, or a DNA. Genetic material is not enclosed by envelope or the membrane and lies in direct contact with the cytoplasm. So this is a very significant and very important feature of your prokaryotic cell, in which the genetic material or the DNA is not being enclosed by any membrane and lies in the direct contact with the cytoplasm, means lies as such in the cytoplasm. So the naked genetic material lies compactly coiled in the region, and now we call that genetic material as nucleoid. And DNA of the prokaryotic cell, that is single, circular, which does not have histone proteins. So they are just DNA is there, with the genetic material, no proteins are being present, and nucleoid is associated with the mesosomes. As we have already discussed, they are the infolding of the plasma membrane. And another one is plasmid. This is again very important. In addition to the chromosomal or the genomic DNA, many bacteria contain a small circular DNA in the cytoplasm. It is called plasmid. And what is the unique feature of the plasmid? That a plasmid had the genes for the certain unique characters like resistance to the antibodies, because the bacteria is a pathogen. Pathogen means the bacteria may cause some diseases, and that particular disease is going to be disinfected only with the help of the antibodies. So plasmid is a circular DNA and they have a unique characters like resistance to the antibodies. So which type of the bacteria is this? Which type of the particular strain is this, by which we can give the treatment, as we have discussed about the gram positive and the gram negative bacteria. In this image, that will be very clear about the genetic material. So this is a genetic material, that is the DNA, which is single, circular and without any membrane and lying as such in the cytoplasm without the membrane. It is naked, so it is known as a nucleoid or the genopore. And this is a circular extra chromosomal DNA lies along with the genomic DNA, and they have a unique feature. They have a genes which give them a resistance to the antibodies. So this is the genetic material DNA known as the nucleoid, and this circular DNA that is known as the plasmid. Next, again a very important feature is ribosomes. Ribosomes are non-membrane bound organelle. So that's why it is present in the prokaryotes, because in the prokaryotes no membrane bound organelles like mitochondria, endoplasmic reticulum are present. Even the nuclear material, or you can say the genetic material, is without the membrane. So prokaryotic will have all the organelles which are without the membrane, and the ribosomes are the non-membrane bound organelles found attached to the plasma membrane of the cell. And the type of the ribosomes which is present in the prokaryote is 70S. S means that is the unit of the ribosome, that is a sedimentation coefficient, and that is also being known as a Sedberg unit. Sedberg is the name of the scientist who has coined the name ribosome. So S stands for the sedimentation coefficient or the Sedberg unit, and 70S consists of two subunits. The large subunit is of 50S and the small subunit is 30S. तो don't get confused कि दोनों का total 80 बना है और हम इसे 70S कहते हैं. 70S ही है with the large subunit that is 50 and the small subunit that is 30. So what is the main function of the ribosome? They translate mRNA into proteins. So hence known as the protein factories of the cell. So they help in the synthesis of proteins with the help of the RNA. We will discuss later on, and how the particular formation of the protein takes place, that is in the genetic chapter in the 12th class. So here we should know only this one. What is the function of ribosomes? They are the site of the protein synthesis. They are known as the protein factories, and several ribosomes become attached together by the strand of mRNA and form the polyribosomes. Means in polysomes means they will have a group of ribosomes, all are being attached on the mRNA, and that group of ribosomes that is being known as the polysomes or the polyribosomes. The next is the inclusion bodies. They are non-membrane bound structure. As I have told, कि prokaryotic will have all the structure which is without the membrane. freely lying in the cytoplasm in which the reserved material are stored. The prokaryotic cell contains the number of inclusions like number 1 is phosphate granules, cyanophysin granules, glycogen granules, and these granules are basically they are storing the food, and that's why they are being known as a reserve material are stored. Reserve material means the food is being stored, and that is known as the non-membrane bound structures. There is one more body is present, that is a gas vacuoles, which is present in the cyanobacteria, purple and the green photosynthetic bacteria, and these vacuole help the organism in positioning themselves in water. As we know, the bacteria that habitat in the water, and they make them to be positioned कि यहीं बैठा रहे, यहाँ पे ही रुका रहे. This is possible only with the help of the gas vacuole, and that is cyanobacteria का मतलब है blue green algae, purple green bacteria photosynthetic bacteria है, and these vacuoles help them to be stationed, help them to be positioned themselves in the water bodies or in the water current. So inclusions, that is basically that particular non-membrane bound structure in which the food is being stored. Examples are that is phosphate granules, cyanophysin granules, glycogen granules, in which we are having a different forms of the food in the reserve form, in the stored form. So the next lesson will be on the prokaryotic appendages, in which we will discuss about our flagella, pili and fimbriae. Till then, stay home, stay safe, stay healthy. Thank you.